nucleus and the chromosome physical basis of heredity. The physical structures present in the cell which are responsible for the transmission and expression of hereditary characters are the chromosomes present in the nucleus. The nucleus. Robert Brown first discovered a prominent body within the cell in 1833 and termed it as nucleus. The nucleus can be called as the controlling center of the cells as it contains chromosomes and genes which control all activities of an individual cell. Generally, a single nucleus is present in a cell, unenucleate, but in some cases more than one nucleus may be found, multinucleate, as in cenocytes, certain fungi, where nuclear division is not followed by wall formation. The shape of nucleus also varies from spherical, oval to flattened lobe or irregular in shape, depending upon the type of cell. In an active cell the nucleus is large while in a resting cell it is small. On the basis of presence or absence of well-organized nucleus, the organisms have been classified as I. Prokaryotes, the organisms in which the nuclei are not well organized, e.g., viruses, bacteria and blue-green algae. 2. Eukaryotes, the organisms which have well-organized nucleus, e.g., the cells of higher plants. The structure equivalent to the nucleus may be a DNA thread or body. Present in prokaryotes is called a prokaryote or nucleoid. The nucleoid does not have a true chromosome enclosed in a nuclear envelope and does not divide by regular mitosis. The nuclei may be absent in some specialized cells of eukaryotes, e.g., in mature red blood cells of mammals. A nucleus consists of three major parts: the nuclear membrane, nucleolus, and chromosomes. The fluid in which the nucleolus and chromosomes are present and which is enclosed in nuclear membrane is called nucleoplasm. Nuclear membrane, the nuclear boundary of interphase and prophase nuclei is called the nuclear membrane or envelope. It usually breaks down at the end of prophase. The nuclear membrane separates the nucleus from the cytoplasm and functions to facilitate and regulate nucleocytoplasmic interaction. The electron microscope demonstrates that the nuclear membrane consists of a double membrane having two unit membranes. The space between the two unit membranes is called perinuclear membrane and it varies in width. The outer membrane is continuous with the endoplasmic reticulum. The outline of the nuclear membrane is smooth and pores are present in it. These pores appear circular in surface view. At the boundary of these pores, the inner and outer unit membrane is joined. In animal cells, a diaphragm extends across each pia and is known as annulus. In plants this is absent. These pores provide direct contact between the nucleus and the cytoplasm. It is believed that the pores can open and close, but the mechanism is unknown. The nuclear membrane disappears in prophase of mitosis and meiosis and is reformed at the end of the nuclear division. The nuclear membrane breaks down at prophase into vesicles which disperse in the cytoplasm. It is believed that at the time of reformation these old vesicles gather together to form the nuclear membrane. Nucleolus, the cells of higher organisms, has a spherical, colloidal body called nucleolus which is associated with a particular organizing chromosome. A special region called nucleolar organizing region is present in the chromosome to which the nucleolus is associated. The nucleolus disappears during mitosis and meiosis at prophase and reappears at telophase. In certain cases more than one nucleolus is found in a nucleus, but these several nucleoli merge together to form a single large nucleolus. The nucleolus consists of RNA, acidic and basic phosphatase. Dyes, phospholipids and alkaline live. The ultrastructure of nucleolus suggests that there are at least three components present in the nucleolus: I. Granula, composed of dense granules, 2. Fibrils, and 3. Amorphous matrix, surrounding the granula and fibrils. After treatment with RNA-SA, the granula and fibrils disappear, indicating that they mainly consist of RNA. The most important function assigned to the nucleolus is the synthesis of ribosomal RNA. granular glutate, and regulate the nucleoglyphtore. Amorphous outer membrane, inner membrane, muttering chromatin, nuclear pore, nucleolus, RNA, acid Bock dyes, phospholy, chromosomes. In the nuclei of higher organisms, definite number of chromosomes are present. Also, the E. Strasbacher in 1875 first discovered thread-like structures named chromosomes. Shape and size is definite.